Alright, let's do this. Okay, cell immunity involves several stages. First up, let's do a quick overview over antigens, pathogens, and antigens. Antigens are simply things that can indicate if are just molecules that can initiate an immune response. Wait, no, they're molecules that Wait, is this private? Just making sure this is private. This is private, right? Yeah, I think this is private. I'm just assuming this is private. Okay, so anyways, antigens are what the flip? Antigens are simply molecules that assist in distinguishing a substance as non-self and can initiate an immune response. Antigens usually are distinguished as non-self because due to cells having MHC1 markers, which immune cells can like identify through receptors that can bind to these markers. Immune cells such as immune cells that are APCs, antigen presenting cells, have MHC-II markers which can present antigens they have obtained from phagocytosis. Pathogens are simply agents or organisms that cause disease to the body. These can include cellular and non-cellular. Cellular involves bacterial, fungal, parasitical, protozoans. Non-cellular involves prions and viruses. Bacteria is the main one, which I'll be tested on. So, yeah. Good thing no one's watching this. Bacteria pathogens are simply bacteria that are simply bacterium that cause illness, cause disease. They come in two types. They could be these guys usually produce toxins which damage the body, because how else would they damage? They don't eat cells, so mainly through production of what do you call it, toxins. There's two types as well: gram A and gram B. Gram A and gram B, I think that's what they're called. Gram positive and gram negative bacteria. Gram positive is simply bacteria that have gram positive are simply bacteria that secrete toxins while they're still living, whilst gram negative are like the bacteria that only release their toxins when they die. For example, Medionosis is bacterium is gram positive because it releases toxins while it's living. These toxins can cause bad stuff to occur, usually kills cells, causes inflammation, and provokes an immune response. That's basically all I know here, because why do I need to know more stuff? It won't be tested, I think. I'm assuming. Subdivisions of immunity. These involve innate immune response and adaptive immune response. Those are the only two major parts. The innate immune response differs from the adaptive immune response since it's basically it is basically immediate, whilst the adaptive immune response requires at least a few days to get kicking, and it's also not as strong as the adaptive immune system. It also does not have memory, unlike the adaptive immune system. Innate immune system also has a couple other branches. It's got the what like the like the cells doing stuff, and then you've got like the cell environment doing stuff. And what's it called again? Sorry, like, I know why it's cell medium humor. That's fucking what? Where is it? Is it just gonna skip what? I guess anyways. There's no also yeah, there's lines of defenses first, second, and third. First line defense are basically barriers preventing entry, such as, for example, physical barriers. Intact skin prevents bacteria from entering the body there. Mucous membranes lining the and airways and nasal cavities. They can trap bacterium or other pathogens attempting to breach the body and are expelled through sneezing and coughing. Chemical barriers, you've got like the lysozymes in like the mucus membranes, the tears, and the saliva that can kill pathogens, And microbiota barrier, which is basically just competition for the bacteria, like they just like take up all their resources and stuff, easy stuff like that. Thank God no one's watching this. Just move down T because I still do not know. Okay. Oh, this is in-depth analysis. Physical barriers. Oh, no, physical barriers. Obviously, oh no, just like tissue that prevents pathogens from crossing body surfaces. I just totally did not look directly there. No, so direct definition, they're just epithelial tissues that prevent pathogens from entering the body. And chemical barriers are they just destroy pathogens on the outer body surface. Nothing too out of the ordinary. Microbiota, that's just does microbiota stuff. Or plants also have the defenses, believe it or not. Physical barriers in plants, you've got the waxy cuticles covering the leaves. These basically prevent moisture from gathering and prevents like pathogens from settling on the leaf surface. The stomata also acts as a physical barrier for the what they call it pathogens, since they cannot enter the leaf inside of the leaf due to the stomata being able to close. Easy as that. During infection, the plant will create a gall, which is basically just tries to like prevent more the infection from spreading up out of like the infection site. What is thorns and spikes doing here? Oh, well, I guess it's that. Chemical barriers. These are just chemicals that kill pathogens. Well, I need to try to kill pathogens and prevent them from entering the plant. Yeah, we've got the usual saponin. These are just proteins that disrupt the bacterial and fungal cell membranes. Only those two, by the way. Caffeine is just toxic to funguses and insects. So yeah, we're just drinking toxins. Helps stimulate us. And then the last one that's probably useful is tannins, which is just disgusting substance that like grazers don't like. Well, that was quick. Damn. Easy peasy lemon squeezy. Components of innate immunity. Oh my god, there's so much. I don't even know what the groups are called. Actually, I actually didn't want to look at this. Immune cells, complement proteins, and inflammation. We're to look at that. So cells involved in innate immunity. These are called leukocytes. Well, leukocytes are just a general term for immune cells. The ones we're looking at are phagocytes, eosinophils, and mast cells. Those are the main three. Oh, and also natural killer cells. For phagocytes, they're just like cells that like eat stuff through phagocytosis. It is basically just cells that are that. What is? How did I say? How do you use phagocytosis in a sentence? These are the cells involved in phagocytosis, basically. For the examples of these are like neutrophils. They're just like the main foot soldier of the immune system because they're just like the standard stocks guy, and they die a lot. Kyrgyzak told me that. Yeah. Some phagocytes are actually, there's a subgroup of phagocytes called antigen presenting cells. These are just phagocytes that can present antigens on their surface after they've engulfed and digested what they call a pathogen. It presents the leftover protein fragment on the MHC-II markers on top of its surface, and they like migrate to lymph nodes to like present these antigens to like activate the adaptive immune response, basically. Monocytes don't need to know that. Eosinophils are just like guys that produce toxins. So yeah, give the bacteria on like a taste of their own medicine. Well, I've got no idea why it says assassins here, because like they just kill infected cells through like releasing perforin via degranulation and like releasing granzymes, which enter through the pores created by the perforin and like initiate apoptosis through that. Yeah, I'm wrong. Word from word here. Yeah, mast cells, they're just like they're cells which like antibodies would bind to, and like they would like, and these antibodies which can detect like the antigen would just like would like activate the mast cell if the antigen binds to them, and like the mast cell would just release histamines. Simple as that. Phagocytosis. Oh, well, I'm gonna try to do this myself. So, pathogen is identified through the lack of an MHC-I marker, so it is declared non-self. Number 2, phagocyte engulfs the pathogen. Stage step 3. The pathogen is also is mixed with lysosomes, which digest the pathogen. And stage 4 is that the cell expels the leftover protein fragments. Yeah, good enough. Would you look at that? I might need to know what phagosome is, but like other than that, that's pretty good. Yeah. This is mainly histamine. You don't need to know about heparin and cytokines and stuff. Stupid. Oh yeah, boss cells are basically just like, like only thing you would like answer it for in a VCE question is legit for antigens. Like you can link them to inflammation, where you could just say that they release histamine. So yeah, detect mast cells detect the what they call a pathogen, release histamines. That's what we need for inflammation. For antigen, you can just say like the antibodies. And the antibodies again that are produced by plasma cells against the antigen are like bind to like the mast cell. Well, at least bind to like the mast cell's receptors, and these are always IgE antibodies. Don't ask me why, because I don't know. So yeah, basically mast cells become like literal like landmines for like the antigen. So as soon as one antigen lands on it, boom, histamine release. And boom, inflammation. Blah, 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 blah. Yeah, and this is like one-time use. So yeah, I mean, like this is like the like one-time thing. You only have to like attach the antibodies one time, because like the antibodies stay there for like much longer. So yeah, fine. Oh yeah, so now we get to the complement proteins. Complement proteins come in 3 flavors: lysis, ones that cause lysis, ones that coat pathogens, and ones that attract leukocytes. So going to lysis, the complement proteins will just like identify that the pathogen is non-self, and they'll just bind to it. They'll like create this like protein complex that like binds to like the pathogen, and like just kills it through lysis. This is not effective against like all stuff basically as well. Coating is basically just opsonization right here, because it just covers the surface of the pathogen and like makes them more recognizable basically. recognizable to like the phagocytes, and so they just eat it. Yum nom nom, So, or chemotaxis, fancy way of saying attracting phagocytes. This is legit what the complement like proteins can do. They just like like act as markers for like the pathogen once they attach to it, and like they just like cause chemotaxis. Well, the last one, interferons. I almost forgot this. This is like virus and like infected cell exclusive because it requires the infected cell. These are just like cytokines released from from an infected cell, and these like warn nearby cells of the viral infection, and this causes the nearby cells to like try and resist the viral infection through either just like destroying all the RNA in their bodies, so like to ensure that any viral RNA inside of them will not be like replicated. They also involves thickening the cell membrane so the viruses can't even penetrate it. And of course, it's like wait, let me just like check. Oh yeah, it activates. Yeah, it's another chemotaxis. Chemotaxis, and it causes the infected cell to kill themselves. So yeah, it released and then they kill themselves. This is a very good one. Signal change. Plasma membrane make entry to the virus much more difficult. Nearby infected cells also undergo apoptosis, and immune cells, specifically the natural killer cells, just go in, kill them. Inflammation is on the next one because this is a long topic. Inflammatory response is 3 stages. You've got like what I don't know what the name of the first one is, or it's vascular stage, cellular stage, and resolution stage. Vascular involves blood vessels mainly, and like cellular stage involves immune cells, and resolution stage, which you believe it or not, is the resolution of inflammation. Simple as that. So there's 4 main symptoms of inflammation, and those are redness, swelling, pain, and heat. This vascular stage causes the swelling, the heat, and the redness, and in turn the pain. So yeah, it just causes all 4 of them. So the reason for the redness is because vasodilation of the blood cells, increased blood flow to that area, makes them more blood, more redness. This is also due to this is also due to exudate being exuded. Exudate is exuded from the blood vessels. I mean capillaries, because like blood vessels cannot all do this. So, like exudate is exuded from the capillaries, and it basically just makes the infected region like swell up, and this puts pressure on the surrounding tissue, which, guess what, causes pain. So yeah, also the increased blood flow also causes more heating because the exudate also contains like clotting agents, but this doesn't matter for viruses. So yeah, that's just cuts and bruises. Basically, exudate pretty much like makes a battleground for the phagocytes. The leukocytes. Cellular stage involves leukocytes squeezing through the capillaries to the infection site, and they just kill nearby pathogens and blah 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 blah. Okay, resolution stage growth. Cytokines cytokines are released throughout the inflammation to make sure the inflammation continues occurring, and once the infection is eradicated, the cytokines that cause inflammation are just slowly released tests, such as it's like rehabilitation for a drug addict, And I also think it's important to mention how inflammation occurs. So basically, infected cells get infected by pathogen. Cells release cytokines. Mast cells nearby probably are stimulated by this in some way. well, like the mast cells probably have like their antibodies already primed to like know if this occurs, and they're just like, oh shit, this guy's attacking us. So yeah, they release histamines as well. So histamines occur, cytokines produced, yeah, boom, inflammation. Lymphatic system. Primary organs, there's two types. Primary lymphatic organs, primary lymphoid organs, and secondary lymphoid organs. Also the lymph, which is the fluid, and the lymphatic vessels, which is where the lymph travels through. Lymph is legit just like a fluid in the lymphatic system that I think, and there's like, yeah, there's one-way flow. Primary lymphoid organs are just like only like the bone marrow and the thymus. Bone marrow is where all leukocytes stem from. Sudden maturation of B cells. T cells migrate to the thymus where they mature. So yeah, those are the primary lymphoid organs. Secondary lymphoid organs, you've got the spleen and the lymph nodes. Spleen, less important of the two. It's just the organ found in like that like filters blood of pathogens due to the presence of phagocytes there, as well as T and B cells. And it removes worn out red blood cells. Easy peasy and squeezy. Lymph nodes are the important part. This is where like antigen recognition occurs. It is where the adaptive immune response is basically initiated. So basically, it's like the link between the innate and adaptive immune systems, as APCs would travel here to present their antigens to naive Th cells and naive B cells, which in turn basically just activate the immune system. I mean, adaptive immune system. That's all you need to know. Would you look at that initiation of adaptive immune response? Okay, so it all steps begins from a dendritic cell or macrophage engulfing a pathogen and digesting it via phagocytosis. As a result, since it's an APC, it presents the leftover protein fragments on its MHC-II molecules as antigens. These like the dendritic cell or a macrophage migrates to travels to the lymph nodes and searches for a naive helper T cell, Th cell with a T cell receptor complementary to the antigen, and as a result, the Th cell binds to it and it is activated, and it undergoes clonal expansion. This results in the production of a ton of Th cells as well as some memory Th cells, which are important for future responses. The Th cells then let's say they are going to activate the humoral adaptive system. They and the APC go and locate a naive B cell with a antibody or B cell receptor, which is complementary to the antigen. So boom, binds to it, and boom, the B cell binds to it, and then the cytokine releases. What is this? Is it? What? This isn't it? What? But it doesn't matter. So the B cell receptor binds to antigen. The B cell is activated, and with the help of cytokines released by the Th cell, and it undergoes clonal expansion, produces a ton of clones, and these clones differentiate into either plasma cell or a memory B cell. Yeah, boom, they spam antibodies and still mediated. This is T cells now. So in the lymph nodes, there are immature CT cells, and the Th cells release interleukins, which stimulate them to like seek for and the so yes, stimulate them, and when an immature started CT cell binds to either an abnormal MHC1 marker of an infected cell, or the antigen presented by the ABC, it will like be activated, and with the help of cytokines released by the Th cell undergoing clonal expansion and differentiate into mostly CT cells and some into memory T cells. Adaptive immune systems is important due to how specific it is to different viral infections and how it's able to produce a faster and larger response in subsequent, like what they call it, subsequent exposures to the pathogen. Two branches: humoral. Humoral is basically just like substance immunity, involves substances. Cellular is immunity caused by immunity through cells. Bing bang, ching chang, bing bang, ooh ee, ching chang, bing bang. Ching chang, bing bang, ooh ee, ching chang, bing bang. Okay, so first up, you've got humoral adaptive immunity. So let's just first check if I was correct in that how it was activated. Yes, HTH cells bound to the antigen with these cytokines. Wait, they're also bound to it? Oh, okay. So the ABC presents the antigen to the B cell, and which has like the B cell with the complementary B cell receptor binds to it, and the Th cell bound to the antigen also releases cytokines which activate the correct B cells. Which activates the B cell that could bind to the antigen. So yeah, differentiate into plasma cells and some LED cells. Okay. So how does the humoral immune system work? It relies a lot on plasma cells spamming out antibodies. Antibodies come in basically Y-shaped molecules consisting of a long chain and a light chain and a heavy chain. Light chain is the two small chains, and like the heavy chain is the two long chains. It consists of a constant region, which is the region that stays constant for all antibodies, and a variable region which changes depending on the pathogen. The variable region is also the antigen binding site. So yeah, good thing. There's also disulfide bonds holding the antibody together. The antibodies bind to the epitope of the antigen, which is basically the part of the antigen that the antibodies can bind to. They have several functions. One is neutralization, which basically neutralizes the pathogen or toxin so that the pathogen, usually virus, cannot like infect body cells, and the toxin is basically inactivated, rendered useless. Opsonization is another one. So yeah, this is where like the antibodies bind to the antigens on the pathogen, producing antibody-antigen complexes, which basically make the pathogen a target for phagocytes and complement proteins, which usually results in the pathogen dying. Precipitation and agglutination, they're basically the same thing. Precipitation is just the antibodies binding to the soluble antigens, making them insoluble, causing them to clump together and making the pathogen more recognizable to the immune system. And agglutination is when the antibodies just bind to the surface of antigens, which create antigen antibody complexes. This causes like pathogens to like possibly clump together, like the whole pathogen in this case, and be more recognizable. Antibodies could also trigger the release of histamine through the binding to more cells. Yeah, so that's that. This is now cell mediated. Yeah, so cell media interested in the threats. Any like cell that does not have MHC1 marker or has an abnormal MHC1 marker, pew, dead. These are usually infected cells and tumor cells. So the process is of how they are, like what they call it, how they kill stuff is like rather different than usual. So it's very similar to natural killer cells. Like bind to the abnormal MHC1 marker on the infected cells, as tumor cell, whatever, and they release performance through degranulation, and then they release granzymes which enter the cell and initiate apoptosis. Boom bang ba 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 ching chang whatever bing bang ooh ching chang whatever bing bang ooh. Natural and artificial immunity. Oh yeah, baby. I'm fucking using it. All right, active immunity is basically the body produces its own antibodies, and it results in the production of memory cells, leading to long-term immunity. Passive immunity, the body does not produce its own antibodies; instead, it receives them from another organism. This results in no memory cells being produced, and as a result, short-term immunity. Natural is where the immunity occurs naturally without like outside intervention, and artificial is where immunity is induced through artificial intervention, external intervention. Okay. I'm gonna pull up another document that you guys cannot see because, like, I don't know, I'm too lazy to change it. This slide is about vaccinations. They are natural active immunity. I mean, artificial active immunity because you're deliberately introducing because it is the deliberate introduction of a pathogen, of like a weakened pathogen or dead pathogen into the body, which provoked an immune response. And basically just activates the immune system without causing disease. An old way to say that. There are 5 types, but only 2 or maybe 3 are important. You've got the naive attenuated vaccine. A naive attenuated vaccine basically contains the pathogen that has been weakened or like just scuffed. So yeah, they're just weakened viruses and bacteria. They can not cause disease, but they can reproduce. This has some dangers, even though this could like mutate or revert to like the viral version of the pathogen, or it could like say like the body is just like extremely weak, like so weak it can't even fight a vaccine. Yeah, then like the man could probably die. But it's better than normal vaccines because it stimulates a higher immune response and it results in longer lasting level of antibodies and therefore immunity to the pathogen. Inactivated vaccines are the next important one. These are just dead pathogens, just injecting them into there. Not as like long lasting as attenuated vaccines, but like still pretty good. mRNA vaccines probably don't need to know about that. It's just very complicated. I'm pretty sure that's the COVID-19 one because, like, yeah. Okay, this is the next probably most important one. The subunit vaccines or protein vaccines, they just contain the antigen and just inject the antigen in there, provokes immune response, blah blah blah. Done. Okay, this is now stuff that is more strange. But I basically no way of saying I did not know much of this stuff. Herd immunity. Actually, I know herd immunity. Anyone knows herd immunity. This is where the majority of a population, or not like the high percentage of a population, is immunized or is immune against a pathogen in an area, which results in non-immunized individuals being protected from the virus. I mean, from the pathogen, since it cannot spread as easily due to not enough, due to like basically like the infectious the virus, I mean the pathogen being unable to infect most people in the population, so it can't spread as far or be transmitted as far, basically. Okay. This is how pathogens are identified. The only one I need to know is immunological, because it contains ELISA, whatever it's called. I like to call it ELISA. Yeah, so ELISA is basically just a serum-based. It's just like detecting the disease through antibodies binding to like the antigen, and yeah. Okay, ELISA, only one I need to know, sandwich method, because sandwiches are cool. Basically, capture antibody, which can bind to the antigen, is like coated to the bottom of a well, and then the excess is washed. Then like the antigen is added, binds to like the capture antibody. Excess is washed. Then an enzyme linked antibody is like it like binds to the antigen, and the excess is washed, and then the enzyme binds to the enzyme linked antibody, causes a kind of change, and boom, yeah. Basically, it ensures that like samples with the disease are turned colored, and samples without the disease do not undergo color change. Mode of transmission, do not need to know that because I'm too smart for it. Scientific strategies are just evidence-based interventions that actually reduce or stop the transmission and infection of a particular pathogen, like vaccines, disinfectants, washing hands, being tested. Social strategies are just policies that affect groups of people and are put in place to support the scientific strategies to ensure they're effectively implemented. Yeah, that's it. Yeah, where's the end stream button? I do not want to do this anymore.